Hello, my name is Elliott.
Hi, my name is Paula Aguirre, and the title of our research project is an investigation of endothelial cell attachment and proliferation on porous polycaprolactone scaffolds for bone tissue engineering. As it is known, bones are essential because they protect the organs, they allow movement, they make blood cells, and have many other important functions for the human body. However, while bones can naturally regenerate, it is to a certain extent. When there is a big defect or poor blood supply, they usually need medical intervention. And traditionally, bone allografts are the gold standard, which is when they take tissue from one part of your body and then they use that tissue to heal the bone defect. But this comes with major issues like pain at the donor site, multiple surgeries needed in the human body. There's a high infection risk and a limited supply, which is why this bone tissue engineering study is so promising because it focuses on using the body's own healing ability to regenerate tissue. We use biomaterial scaffolds, and they not only provide structure, but they also support cell growth and they deliver bioactive factors to the area of the bone defect. Um, for scaffolds, we use polycaprolactone because it is a strong but also flexible material, and it is biocompatible and biodegradable, which is something we look for. Um, and the cell line we use in this study is the human umbilical vein endothelial cells, the HUVIC cells, since they promote angiogenesis, which is critical for bone repair. In the table to the bottom of the introduction section, we can see what we're testing in the scaffold. We're testing the porosity, which is how many holes the scaffold has, and we have a 70% porosity, 80% porosity, and 90% porosity. We also test the porogen size, which is how big or small the holes are in the scaffold, and we have a 106 to 300 micrometer, 300 to 500 micrometers, and then 500 to 710 micrometer. We also test different coatings on the scaffold. For control, we use endothelial media, and we also have hyaluronic acid, which helps with cell attachment to the scaffolds. And we also chose to use gelatin because it is a natural, biocompatible, and biodegradable um, environment that it gives to the scaffolds. For a hypothesis, we hypothesize that the porosity, origin sizes, and types of coating will have different effects on the cell proliferation of cubic cells. And our objective is to investigate the different effects of the origin sizes, the porosities, and the coatings on that they're gonna have on the endothelial cell attachment and proliferation. For our methods section, for part one, we have the fabrication of the scaffolds, and we first measure out the polycaprolactone, then we dissolve it in dichloromethane, and then we add the salts to create those pores, and then we let it dry, and then we punch out the scaffolds with an 8 millimeter diameter times 1 millimeter height. And then we use a solvent casting method to leach out the salt from the scaffolds, so the scaffolds are left with the holes in them. And then lastly, we lyophilize them in order to freeze dry them. And then for part two, or for our methods section, we have the actual cellularity study, the MTTSA, and then we first culture HUVIC and we grow them to an 80% confluency, and then we seed the cells into the scaffolds. Then we do the MTTSA, which is added to each scaffold, and then after the formicine crystals are formed, we dissolve them with dimethyl sulfoxide, and then we read the plate with a microplate reader at a wavelength of 570 nanometers.
So, for our results, our first part is the scaffolds themselves. So we can see an example in figure 1 of an 80% porosity scaffold. The bottom left image actually shows what they look like. They basically are little circles that almost have a spongy texture, but they're still pretty tough. And to the right of that image is actually what these scaffolds look like when they have the formosyn crystals growing on them. And in figure 2, we can actually see what they look like under a scanning electron microscopy. And what this best shows us is the difference in surface areas when it comes to our porosity and porogen sizes. So, for example, when we're looking at porosity, our lowest surface area can be found in our 70%, and our highest can be found in our 90s. And why this matters is scaffolds with a higher surface area, there is more place for the cells to adhere to, so it leads to improved attachment. But it comes at the cost of not only are these scaffolds a bit more delicate, but they also degrade at a quicker pace. And in contrast, our lower surface area scaffolds like these 70s, they're definitely a bit tougher, but they're less flexible, and there's less places for the cells to adhere to. So depending on the injection site, these details can really matter, especially when it comes to the rate of degradation, depending on the injury that's being treated. And then for our next section, it is the cells themselves, and we're starting with porosity and porogen sizes. On the left is an older study that we did at and it is labeled as our 0.01% concentration. And on the right is a more recent study that we did labeled as 0.1% concentration. And the only difference between these two studies is how the coatings were made. So there was no difference between porosity and porogen sizes when testing. And when we're looking at this data, these graphs can tell us a few different things. What day 1 tells us is the cell's rate of attachment. So basically, how well could the cells attach? How hospitable is the environment? And then looking at day 7, we are looking at the cell's abilities to proliferate, as well as the cell population as a whole. And it's important to note that while it may look like, just based off a glance, that the data may look like it's trending down or up, we actually can only come to conclusions when we see statistical significance. And the only instance of statistical significance is what we see in our 0.01% concentration study. From day 1 to day 7, we see a significant decrease in cell number in scaffolds that are of a porogen size of 106 to 300 micrometers. And then coming to our coatings, this is where that difference in concentration is. On the left is a lower concentration, on the right is a higher concentration. And what I mean is just the ratio of gelatin to water or hyaluronic acid to water. And so interestingly, we can see on the left that our gelatin showed to be significantly higher than the hyaluronic acid in control on our day 7. And that basically tells us that it's a more hospitable environment for our Huvic cells because they had an easier time not only maintaining cell population, so less cells died, but also they had some capacity to reproduce. And interestingly, we found on the right, at higher concentrations, not only did we see that same trend of on day 7, our gelatin being significantly higher, seeing that significant increase in population, but also we saw a higher rate of attachment, meaning when compared between the control group and our gelatin. And what this tells us is that something in the gelatin is allowing for the Huvex to more easily attach. And what it actually is gelatin has something called the RGD sequence. And this sequence is able to more easily bind to the integrants found on the surface of Huvex cells, allowing for easier attachment and ultimately higher cell populations at a later date. So with all of this data in mind, we concluded that while our Huvic cells really showed no particular preference for porosity or porogen size, they showed a pretty strong preference for gelatin coating. And in the future, we are planning to repeat this study, doing gelatin-coated scaffolds in different porosities and porogen sizes. But yeah, thank you.
Thank you. Okay.